Hello, my name is Dr. Danny Meyersfeld, and I'm going to be speaking to you this evening on the question of complementary alleles. This will be the first lecture in a series of talks that we'll do to assist the healthcare practitioner with the interpretation of genetic reports. The goal within our organization is to see genetic testing form part of routine clinical practice. And none of us are so naive as to think that it is only our tests that will ever be used by all practitioners. As a company dedicated to education of healthcare practitioners, we think it's important that these practitioners find comfort levels with all formats of report, and that any and all genetic reports that end up on a patient's or on a doctor's desk can be understood by that doctor. The question of complementary alleles is one that comes up quite often, so I'm going to take just a few minutes to unpack this and try and get to the bottom of what might appear to be a discrepancy between reports, but it is in actual fact quite common. The unfortunate thing about the field of nutrigenomics is that there is an obvious lack of consistency between different testing companies with the manner in which the information is presented. And this is what has led to the question of complementary alleles, which you'll understand better in a few minutes. What we're seeing here is what is commonly known as the central dogma of biology. A double-stranded DNA molecule, through a process of transcription, is converted or copied into a single-stranded RNA molecule, which through a process of translation is then converted into a chain of amino acids that functions as a protein. This is an unbelievably complex process that is happening in all of our cells all of the time, and it's really quite astounding when one sees animated images and videos of this process to think of the complex molecular machinery that is actually happening in our cells. And I would encourage you to try and find some images and some videos of this process if you can. It's really quite fascinating to see. So I've asked the question before, why does a DNA molecule need to be double-stranded? And one of the answers that seems to be most apparent is that it's really a protection mechanism, that the message enclosed within the DNA molecule, these strings of nucleotides known as adenine, thymine, cytosine, and guanine, that message is so critical to the functioning of a cell, and the reliable replication of this message without errors or risks for damage is so important to future generations of cells, that this molecule exists in a double-stranded format so that the sugar phosphate backbone can really protect the message that is enclosed within it. The image that you're seeing clearly shows the pairing mechanism of these nucleotides, where, as you know, a cytosine and a guanine can only ever pair with each other, and an adenine and thymine can only ever pair with each other. And because these molecules are slightly different structure, slightly different sizes, the pairing mechanism that they have creates this helical structure of the DNA molecule. Now, getting on to complementary alleles, we all know that a SNP is a change in a nucleotide between two individuals. So what you can see is person 1 and person 2, where their SNP exists within the DNA. Person 1 has the A nucleotide at a particular position, and person 2 has the G nucleotide. Now, because of the mechanism of pairing, the A nucleotide automatically has to pair with a T, with a thymine, and the G nucleotide automatically has to pair with a cytosine. So you can ask the question, is this SNP an A to a G change, or is it a T to a C change? Which allele do we report? And this is where there is a lack of consistency in the literature. Just going back to the central dogma of biology in order to look at the naming of the two DNA strands. Here you've got the double-stranded DNA molecule, which through a process of transcription is copied into that single-stranded RNA molecule that I mentioned earlier. The strand that is copied is called the template strand, and that will have the opposite message or the complementary message to the eventual RNA molecule. The other strand, the strand opposite this, is known as the coding strand, as that strand will have the identical message as the eventual RNA molecule. Now, the coding strand is also called the forward strand or the plus strand in some cases. So one of the options for the naming of the alleles is to always refer to the nucleotide that appears on the forward strand. So whichever nucleotide is appearing on the forward strand, that is how we would name the SNP. The problem with that, and the reason that we have chosen not to follow that nomenclature, is one of the most commonly known SNPs that we work with, the MTHFR 677C to T. That SNP actually appears on the reverse strand, or is named on the reverse strand. And so if we were to follow the convention of naming according to the forward strand, we would actually have to refer to that as MTHFR 677GA. Now, if you go to the literature and do a search on MTHFR 677GA, nothing is going to come up because that is not how it's named in the literature. And so that gets us to the convention that we have decided to follow, is because of the manner in which we encourage practitioners to refer to the literature all the time, to do their own reading, to go and look up the papers and find the articles on the different SNPs that we look at, we've decided to name the SNPs according to the manner in which they're named in the literature. This means that sometimes they'll be named according to the forward strand and sometimes they'll be named according to the reverse strand, but it does make it easier to identify the SNPs when you do those literature searches and look for articles. So a quick test to see if you've learned anything from this short webinar. If you see the letter BDNF or the gene BDNF and one report has a CC and report 2 has a GG, that is not a problem because those are complementary alleles. Similarly, if MTHFR 677 in one report is referred to as a CC, whereas another report is referred to as a GG, again, these are complementary alleles and so that would not be an issue. If, however, report 1 has a result for IL1A as CC, the homozygote, whereas report 2 shows CT, the heterozygote, that would clearly indicate that there has been a lab error or reporting error somewhere along the line. And in that case, I would encourage you to seek further explanation from the laboratory to understand which genotype is correct. Just to take a look inside the DNA laboratory, it is a SANUS accredited laboratory according to standard ISO 15189-2012. With regards to quality control, we run positive controls, we run negative controls, we do random duplicates in every run. We also do interlab proficiency testing with 2 international labs. We have a lab in Helsinki and a lab in the US, and we continually exchange samples between these labs to ensure that the lab minimizes the risk for error. We claim a 99% genotyping accuracy. Any lab that claims that genotyping is 100% accurate is not being honest. The unfortunate reality is that genotyping can have errors, be it human error or the quality of the DNA that is received by the lab. You do get genotyping errors. What is important is to be able to recognize in the report, is this a mistake from the laboratory or is it simply a being reported in a different way? So in summary, use the reports with which you feel comfortable and feel comfortable to interpret them all equally. Recognize the reason for difference in style and nomenclature of different reports. Question the discrepancies that don't make sense. And importantly, understand where the DNA result fits into the clinical decision-making process. You as the doctor have far more information at your disposal about your patient than we do as the laboratory. Our job is most certainly to provide you with accurate genetic results that can guide you in a particular direction, but we certainly can't be telling you exactly what to do based on the results of just a genetic test. Thank you for your time, and I look forward to having further discussions with you in the future.